Hi everyone, welcome to our session. Today we are discussing about density gradient centrifugation. Myself Anjali PT, second MSc Biology, St. Mary's College, Thrissur. First we are telling about what is centrifugation. Centrifugation is a technique of separating substance which involve the application of centrifugal force. The particles are separated from a solution according to their size, shape, density, the viscosity of the medium, and rotor speed. More simply, centrifugation is the technique of separating components where the centrifugal force acceleration causes the denser particle or substance to move towards the periphery and less dense particle move to the center. The rate of centrifugation is dependent on the size, density of the particle present in the solution. Then we are discussing about density gradient centrifugation. This type of centrifugation is mainly used to purify viruses, ribosomes, membranes, etc. A sucrose density gradient is created by gently overlaying lower concentration of sucrose on higher concentration in centrifuge tubes. The particles of interest are placed on top of the gradient and centrifuge in ultracentrifuges. The particles travel through the gradient until they reach a point at which their density matches the density of surrounding sucrose. The fraction is moved and analyzed. It is two classes: rate zonal density gradient centrifugation and isopanic centrifugation. Once more I am telling, density gradient centrifugations are two: rate zonal density gradient centrifugation, isopanic centrifugation. Rate zonal density gradient centrifugation. Zonal centrifugation is also known as band or gradient centrifugation. It relies on the concept of sedimentation coefficient, movement of sediment through the liquid medium. In this technique, density gradient is created in a test tube with sucrose and high density at the bottom. The sample of protein is placed on the top of the gradient and then centrifuged. After centrifugation, faster sedimentation particles in sample move ahead of slower ones, sample separated as zones in the gradient. The protein sediment according to their sedimentation coefficient and the fractions are collected by creating a hole at the bottom of the tube. The density gradient does not change very much down the tube. Then isopanic centrifugation. The sample is loaded into the tube with a gradient forming solution on the top of or below preformed gradient or mixed with self-forming gradient. The solution of the biological sample and cesium salt is uniformly distributed in a centrifuge tube and rotated in an ultracentrifuge. Under the influence of centrifugal force, the cesium salt redistribute to form a density gradient from top to bottom. Particles move to point where their buoyant density equal that part of gradient and form bands. They form bands. This is to say the sample molecules to move to the region where their density equal the density of gradient. It is a true equilibrium procedure since depends on buoyant density, not velocity. Example, cesium chloride sodium gradients for macromolecules and nucleotides, self-forming gradients under centrifugal force, requires steep density gradient. During centrifugation, once a particle reaches a part of the solution with the same density as itself, it will not sink further. Then, applications of centrifugation. To separate two miscible substances and to analyze the hydrodynamic properties of macromolecules, and purification of mammalian cells, and fractionation of subcellular organelles, fractionation of membrane vesicles, separating chalk powder from water, then removing fat from milk to produce skimmed milk, separating particle from an airflow using cyclonic separation, the clarification and stabilization of wine to separate of urine components and blood components in forensic and research laboratories, and aids in the separation of proteins using purification and techniques such as salt out, example ammonium sulfate precipitation. These are applications of centrifugation. Thank you for watching this video.